My name is Sarah Gaffen, and I'm from the University of Pittsburgh, and I'm pleased today to share with you results from our new JEM paper describing the role of a non-canonical RNA binding protein, Arid5a, in driving IL-17-dependent autoantibody-induced glomerulonephritis. This work was led by Yang Li with a variety of wonderful collaborators listed here. The take-home message of this paper is that the cytokine IL-17 drives pathology in autoimmune kidney disease through regulation of a RNA binding protein, Arid5a, which is induced during disease in the kidney, interacts with the ribosomes, and particularly the small ribosome subunit, to induce translation of a variety of targets. These include CAT enhancer binding proteins, which are essential for inducing expression of genes that underlie pathology. My lab broadly is interested in IL-17 signaling and how it pertains to autoimmune disease. And we know that blocking IL-17 is very effective in a number of conditions, such as psoriasis and psoriatic arthritis. There's interest in targeting these to other diseases as well, but very little is known about this in the context of kidney disease. Chronic kidney disease afflicts as many as 15% of adults in the US, and is often many of whom don't even know they have disease. Many of these are driven by autoantibodies that deposit in the kidney and cause local pathology. Diseases such as lupus nephritis, IgA nephropathy, and others are examples of this kind of condition. And we sought here to determine the role of Arid5a in these conditions. We found in human renal biopsies that not only was IL-17 elevated, which was already known, but Arid5a was as well. Using a mouse preclinical model of disease, we saw the same thing was true. Arid5a was induced markedly in the kidney during disease, and mice that lacked Arid5a were completely refractory to the signs and symptoms of disease. For example, elevated creatinine, which is seen in wild-type mice, but not in Arid5a knockouts, or in mice that can't respond to IL-17. To look at the molecular mechanism of this, because Arid5a was an RNA binding protein, we sought to look transcriptome-wide at the targets of Arid5a in the context of renal epithelial cells. We know from prior work that these are the essential cells that IL-17 signals through in the context of this condition, at least in mice. Doing a RIP-seq analysis of cells treated with IL-17, we found a variety of targets of Arid5a, and among these were the CAT enhancer binding proteins CEBP beta and CBP delta. Interestingly, Arid5a, although it bound to these RNAs, did not change the levels of RNA accumulation. So cells lacking Arid5a still had perfectly normal upregulation of CBP delta, for example, at the RNA level. However, it was a different story at the protein level, where IL-17 induces CBP delta protein, but this was markedly attenuated in cells lacking Arid5a. We showed this in a human cell line and also in primary mouse renal tubular epithelial cells, not shown here. This suggested that the effects were more at the protein level, and indeed, broadly, the RNAs that Arid5a bound to after IL-17 stimulation were related to protein translation, metabolism of RNA, protein elongation, ribosomal RNA processing were all pathways associated with Arid5a occupied genes. And indeed, as one example, we found IL-17 induced marked association of Arid5a with the 18S RNA constituent of the 40S ribosome subunit. After IL-17 treatment, we found that Arid5a was exported from the nucleus and inducibly associated with ribosomes. And this, and as well as a lot of data that I haven't shown you, led to a model in which Arid5a associates dominantly with the 40S subunit and helps to facilitate translation of many proteins, including the CAT enhancer binding proteins. If you want to know more, take a look at the paper. I hope you enjoy it, and maybe you'll come to love IL-17 as much as we do.